Today we will study lecture 5, course BBT315 metabolism. So here we will study all about the enzymes. So enzymes are biological catalysts, and under biologically relevant conditions, uncatalyzed reaction tend to be very slow because most of the biological molecules are quite stable in the natural pH, mild temperature, in aqueous environment inside the cell. So enzymes greatly increase the rate of biological reaction by providing a specific environment within which a reaction can occur more rapidly. Enzyme catalyzed reactions take place within the confine of a pocket on enzyme. This is called active site, and the reactant molecule is referred to as the substrate. The surface of the active site is lined with amino acid residue with substituent group that bind to the substrate and catalyzes chemical transformation. Often the active site encloses the substrate, sequestering it from the solution. লাইক এক্সাম্পল দ্যাট একটা বাকেটের মধ্যে যদি বালি নেওয়া হয় এবং বালির মধ্যে যদি অনেকগুলো আয়রনের টুকরো ছড়িয়ে ছিটিয়ে দেওয়া হয় এবং একটা ম্যাগনেট দিয়ে যদি এই বালির ভিতরে নাড়াচাড়া করা হয়, তাহলে সেই ম্যাগনেটটা কিন্তু আয়রনকে ঠিকই ক্যাপচার করে নেবে। বাট তুমি যদি এখানে অ্যালুমিনিয়াম দাও, এটা কখনোই সে করবে না। এভাবেই আসলে এনজাইম তার সাবস্ট্রেটকে খুঁজে নেয়, লাইক দিস টাইপস অফ স্পেসিফিসিটি. So enzymes are very specific and readily discriminating between substrate with quite similar structure. And some enzymes can only catalyze one chemical reactions. So there are two models of enzyme functions. One is lock and key model, where each enzyme fits exactly on its substrate and complementary shape like a lock and key. And in this fit model, this is the extension of lock and key model, where some enzymes can bond multiple substrate, and interaction between enzyme and substrate induce changes in enzyme to feed it. Enzymes commonly enhance the reaction rate in range of 5 to 17 orders of magnitude, and the rate enhancement come from two distinct but interwoven pairs. The first one is the catalytic functional group on an enzyme react with a substrate and lower the activation energy barrier for the reaction by providing an alternative lower energy reaction path. And secondly, non-covalent binding interaction between substrate and enzyme release a small amount of free energy, which is with each interaction that helps lower the energy of the transition state. And the energy derived from enzyme substrate reaction is called binding energy. And the magnitude of a rate constant is inversely and exponentially related to activation energy. Thus, a lower activation energy means a faster reaction rate. So here this table indicates some rate enhancement produced by enzymes. So enzyme affect reaction rates, not the equilibria. A simple enzymatic reaction looks like that: enzyme plus substrate, enzyme substrate complex, enzyme product complex, enzyme product. The function of a catalyst is to increase the rate of the reaction, and catalysts do not affect the equilibrium. The rate of the reaction depends on the energy barrier, that means the activation energy. A higher activation energy, the slower the reaction. Rate of reactions can be increased with increasing temperature, increasing pressure, or the addition of catalyst. In cellular metabolism, groups of enzymes work together in sequential pathways to carry out a given metabolic process, such as a multi-reaction breakdown of glucose to lactate, or the multi-reaction synthesis of an amino acid from simpler precursor. In such enzyme system, the reaction product of enzyme becomes the substrate to the next. The activities of enzyme are modulated in variety of ways. One is controlling enzyme quantity and controlling enzymatic activity. So we can control the quantity of the enzyme through transcription, translation, or enzyme turnover. And we can also control the activity of enzyme through allosteric regulation, covalent regulation, association and dissociation, proteolytic cleavage of proenzyme. So let's discuss those things more details. So the activities, that means for allosteric regulation, here end products are often inhibitors, and allosteric enzymes functions through reversible non-covalent binding of regulatory compound called allosteric modulators or allosteric effectors, which are generally small metabolites or cofactors. Allosteric modulators binds to the site other than the active site. Other enzymes are regulated by reversible covalent modification. Both classes of regulatory enzymes tend to be multi-subunit proteins, and in some cases the regulatory sites and active sites are on the separate subunit. Metabolic systems have at least two other mechanisms of enzyme regulation. Some enzymes are stimulated or inhibited when they are bound by a separate regulatory protein. Look at here. So here, this is the allosteric site. An allosteric inhibitor will bind there, and it will change the active site so that enzyme will not be able to bind with the substrate. And for allosteric activation, allosteric activator bind there and changes the active site according to the substrate, and thus substrate can bind with the enzyme. Some enzymes are regulated by covalent modification. Here, the covalent modification of one or more of the amino acid residue in the enzymes occur, and it is slower than the allosteric regulation, and it is reversible and requires one enzyme for activation and one enzyme for inactivation. Over 500 different types of covalent modification have been found in the proteins, and commonly modifying groups are phosphoryl groups, acetyl, adenine, etc. So here, this is the most common covalent modification. Involve protein kinase or phosphatase, and protein pyruvate dehydrogenase kinase inactivated by phosphorylation by pyruvate dehydrogenase is inactivated by phosphorylation, and this phosphorylation is mediated by pyruvate dehydrogenase kinase. And here, the pyruvate dehydrogenase inactivated pyruvate dehydrogenase can be activated by pyruvate dehydrogenase phosphatase. That means phosphorylation inactivate pyruvate dehydrogenase, and dephosphorylation activate pyruvate dehydrogenase. So here is a cascade system. You can see the inactivated protein kinase can be activated protein kinase 1. That means inactive protein kinase 1 will be activated and form active protein kinase 1 by relay molecules. It might be hormones or extracellular signaling, and then active protein kinase 1 will activate protein kinase 2. It will activate protein kinase 3, it will activate protein kinase 4, it will activate protein kinase 5, and finally it will activate the target protein, and then cellular response occurs. And when this cascade mechanism has to be stopped, then another enzyme, phosphatase, can inactivate this to this one. So what is the proteolytic cleavage of proenzyme? Proenzyme are precursor enzyme that is functionally inactive, are also called inactive zymogen, like chymotrypsinogen, and proteolytically it will be cleaved here and here to produce this one, the final enzyme. This is functional, like proinsulin. This is the one chain of insulin. When this blue color part is removed, the two chain will be found, and this is the insulin. Then feedback inhibition, this regulates the activity most of the enzyme, and pathways can be regulated by feedback inhibition, where end product of a pathway binds to the allosteric site of the first enzyme, altering its structure of the pathway and shutting down the pathway. When product is abundant, active site are full and enzyme's activity dropped. When product is used up, inhibition is reduced and more product is produced. So here, when the thyroxine or thyroid stimulating, when the thyroid hormone level has been reduced in the blood, it will trigger the formation of thyroid stimulating hormone from hypothalamus. Then it will work in a pituitary gland to produce more thyroid stimulating hormone, and then it will work in the thyroid to produce thyroid hormone. When the concentration of the thyroid hormone is increased, it will inhibit the thyroid stimulating hormone from hypothalamus, like this. So the end product will inhibit the first enzyme from ribose 5-phosphate to 5-phosphoriblosamine. So enzymes are subjected to reversible or irreversible inhibition. Enzymes inhibitor are molecules that interfere with catalysis, slowing or halting enzymatic reactions. For example, aspirin inhibits the enzyme that catalyzes the first step in the synthesis of prostaglandin, compound involved in many processes, including some that produce pain. The study of enzyme inhibitors also has provided valuable information about enzyme mechanism and has helped define some metabolic pathways. There are two broad classes of enzyme inhibitor, reversible or irreversible. So in reversible inhibition, we can categorize it in competitive, non-competitive, and uncompetitive. In competitive, here the inhibitors bind to free enzymes only and competes with substrate, and increasing the concentration of substrate will overcome the inhibition. But here inhibitors bind to free enzyme or enzyme substrate complex, and increasing the substrate cannot overcome the inhibition. And in uncompetitive, here inhibitors bind with the enzyme substrate complex only, and increasing the substrate favor the inhibition. And irreversible inhibition. The irreversible inhibitors bind covalently with or destroy a functional group of an enzyme that is essential for the enzymatic activity, or form a particular stable non-covalent association. An example of an irreversible inhibitor is diisopropyl fluorophosphate, which is present in nerve gas. It binds to the enzyme and stops nerve impulse being transmitted. Another example of where we use irreversible inhibitors in medicine is penicillin. Penicillin works by inhibiting the activity of enzyme responsible for the creation of bacterial cell wall. This means that water can enter the bacterial cell, causing it to swell, burst, and die, termed lysis. That's all from this for this